In 2000, Mylotarg became the world's first antibody-drug conjugate, or ADC, a targeted leukemia treatment designed to latch onto cancer cells and deliver a toxic payload. While it was initially approved, the drug was voluntarily withdrawn in 2010 after a confirmatory clinical trial failed to prove its benefit. However, independent researchers continued studying the drug. They discovered that by using a fractionated dosing regimen, splitting the original heavy dose into 3 smaller portions over the course of a week, they could significantly reduce side effects while maintaining the drug's effectiveness. This discovery prompted an effort to bring Mylotarg back to the market. To relaunch in 2017, the manufacturers had to replace the original 1990s production blueprint with a modernized, high-yield process that met contemporary regulatory standards. This created a highly specific legal requirement. Because the reapproval relied on historical clinical data, the new manufacturing process had to produce a molecular profile identical to the version produced 17 years prior. The team faced a unique constraint. They had to overhaul the entire production pipeline without changing a single characteristic of the final product. The first component of this ADC is the gemtuzumab antibody. During the effort to modernize production, researchers analyzed the antibody's makeup and discovered an unexpected variation in its structure that wasn't fully accounted for in the original records. The root cause was eventually traced to a historical change in a raw material supplier for the cell culture. This change had triggered an unintended amino acid substitution, where different amino acids were being integrated into the antibody structure at multiple sites. This presented a major hurdle for regulators. The development team had to provide definitive evidence that these structural mutations did not interfere with how the antibody binds to its target on leukemia cells. Researchers launched a series of biophysical tests to compare the old and new batches. They looked at everything from binding affinity to cytotoxicity, demonstrating that the substituted antibody performed identically to the original version used in the first clinical trials. By proving that these structural variations were functionally neutral, the team demonstrated that molecular changes in the process don't always alter the drug's clinical profile. The second part of the drug is the payload, calychiamycin. It is an extremely potent toxin that requires specialized handling, and the original method for making it was highly inefficient, requiring several complex chemical steps performed directly on the valuable core molecule. Scientists developed Process 3 to modernize production. This new method eliminated redundant steps and removed the harmful chlorinated solvents used in the original 1990s workflow. However, Process 3 initially struggled with low yields. The team discovered that the payload was getting trapped in a dead-end disulfide state during the step where the linker is attached. This diagram shows the chemical solution. By adding a coupling agent called ADC in blue, scientists forced the dead-end product back into a productive pathway. This single adjustment doubled the yield and stabilized the process, ensuring a consistent supply of the activated payload. By understanding the underlying chemical equilibrium, the team drastically improved the manufacturing efficiency while keeping the payload's final structure untouched. The final assembly phase is called conjugation, where the toxic payload is chemically attached to the antibody to create the completed drug. Analysis of the original Mylotarg batches revealed a strange historical anomaly. Exactly 50% of the antibodies in every dose remained completely empty, carrying 0 payload. This chart highlights the specific challenge. A modern process would typically aim for a tight bell curve, where every antibody carries a similar load. But because the team had to match the original drug profile, they were required to intentionally recreate that 50% empty ratio. The team used complex multivariable mathematics, a method called design of experiments, to map out how temperature and antibody concentration affected the reaction. This allowed them to dial in the precise parameters needed to replicate the original irregular loading profile. The ultimate test of the new engineering was the ability to use high-precision controls to intentionally produce a batch that was as imperfect as the original. The final submission to global health authorities involved intense technical debates, particularly regarding which starting materials were used to build the payload under international safety guidelines. These efforts were successful. In September 2017, the FDA reapproved Mylotarg for the U.S. market, followed by approvals in major markets worldwide. The team had successfully balanced the requirements of 2017 manufacturing science with the need to remain faithful to a clinical profile established at the turn of the millennium. It is a reminder that delivering life-saving medicine relies just as much on mastering the microscopic details of its production as it does on the initial medical discovery.